So now we're going to talk about transcription, which is the process of taking that DNA sequence or DNA code and converting it into an mRNA sequence. So there's three overall stages: the initiation, the elongation, and the termination. So we're going to start obviously with initiation. And what things have to happen in order to initiate transcription? Well, we know that DNA is double-stranded. So the DNA must become unwound, or in other words, those double strands must break apart to form this bubble, okay, so that the RNA polymerase, the enzyme that's going to make this new mRNA transcript, can actually make contact with the template strand. Now, there's a special position on the DNA sequence where the RNA polymerase will bind and will begin the transcription process, and that's called the promoter region. Okay, so this would be what we call upstream of the gene, meaning that it is before the sequence of the gene occurs if you're going in the direction that the polymerase is going to move. Now remember that the DNA is double-stranded, so there will be what we call a template strand. That's the one that the RNA polymerase is actually going to be reading, right? It's going to read one nucleotide at a time and add the appropriate mRNA nucleotide when it reads the template strand. And then there's the non-template strand, which is the opposite, right? The other double-stranded DNA sequence. Now, when you read the sequence of the non-template strand, it's going to read the same as the mRNA transcript that's produced, but the difference is going to be that for every time there was a thymine in the DNA non-template, you'll see a uracil in the mRNA transcript. Now, another thing to consider, and we'll talk about this a little more when we go into gene expression, but there are additional factors or proteins called transcription factors. And when we're talking about a eukaryotic cell, there are certain transcription factors that we call basal transcription factors that are necessary. So in other words, the RNA polymerase enzyme is not going to be able to bind to the promoter and initiate transcription unless these transcription factors are present. Now, in addition to that, there can be repressor and enhancer transcription factors. In other words, enhancers would be promoting the transcription, right? Causing the transcription to happen. The repressors would then be preventing transcription from occurring or preventing that RNA polymerase from binding to the promoter. So in this figure, what we see is in blue is the double-stranded DNA sequence. Notice the one on the top is what we're calling the non-template strand, so it's the one that is not actually being read by the RNA polymerase enzyme. And on the bottom is the template strand. Now this region that's highlighted in red is what we're designating as the promoter region, and notice that it is what we would call upstream or before the sequence of the gene. Now this part that you see here, the transcription bubble, is where the transcription is actually happening. And you can see that this RNA sequence, which we know now is the messenger RNA, is being produced. So we're going to get a close-up look at that in a minute. But for every, let's say if this right here was an adenine and this was a thymine, then this would be one at a time adding the appropriate nucleotide. So for this adenine, there would be a uracil, and for this thymine, the next one added would be an adenine. Now, so we've covered initiation. Now we move to elongation. And in elongation, this simply means one at a time, the individual correct nucleotide is going to be added to that growing mRNA transcript. And this is, of course, carried out by RNA polymerase. And the direction, remember we talk about DNA and RNA in direction 5' to 3' based on the position of those carbons around the sugar. So it's just giving us an idea of the direction of those strands. So the polymerase is going to add nucleotides in the 5' to 3' direction. That is in the new, so the mRNA transcript that's being produced. Okay. Because the strands are running in opposite direction, that means the template strand is being read in the opposite direction, the 3' to 5' direction. Remember also that uracil replaces thymine in the mRNA transcript. So let's have a look at this. So we see that transcription bubble again. We see the action of RNA polymerase, and I want to show you, if you just look at the two DNA strands, okay, and when they are together, we know that they're complementary, right? This is the hydrogen bonding that's occurring that's going to hold those together. And we know that they're anti-parallel, meaning since this one is running in the 5' to 3' direction, this one is running in the opposite direction, right? So this would be 5' to 3' this way, and this is 3' to 5' this direction. Now, the template strand is the one that is 3' to 5', and the new sequence then is 5' to 3', just like the non-template strand. So one at a time, this RNA polymerase is adding the appropriate nucleotide as it reads the DNA template. So you see that these are paired up. There's an adenine added for every thymine. There's a uracil added for every adenine, a guanine for every cytosine, and a cytosine for every guanine. So this is going to be one at a time until we get to some kind of trigger that says this is complete and now we need to terminate transcription. So we've talked about initiation, we've talked about elongation, and now we are going to discuss termination. So termination really means stop, right? We're finished. That's the entire transcript. That's all we need. Now, in prokaryotic cells, there can be a very specific sequence on the template that when the enzyme reads that, it signals stop, you're done. Okay, and that would be like we would call that a stop sequence, right? A termination sequence. There's also a protein called Rho that can initiate or cause the termination of transcription in bacterial cells. In eukaryotes, this is actually more complex, more complicated, and what we just need to understand is there are additional proteins that can signal to the enzyme, to RNA polymerase, that you're finished, it's time to stop and release that mRNA transcript. So what I want us to see here for just a second, I want you to look at this, and I want you to first notice that this green ribbon or green line is actually the template DNA that's being transcribed. Those circles shown in red are ribosomes. And the blue lines are actually the mRNA transcript that's being produced. So I want you to look at this for just a minute and maybe pause the video, and I want you to tell me, what kind of cell is this in? Is this in a eukaryotic cell? Or is this a prokaryotic cell? And how do I know? So pause your video, think about it for a second, and then when you're ready, come back and we'll discuss. So this is actually a prokaryotic cell. And how do I know that? Well, the reason I know that is the DNA is here, right? But ribosomes are also present at the same time. So in a eukaryotic cell, so if we draw a eukaryotic cell down here, we have a nucleus, okay? And in the nucleus is where all of the DNA is at. So that would mean that transcription would be happening there, right? Because that's where the DNA is. But the ribosomes, okay, which they show in blue here, so, I mean in red, excuse me. The ribosomes, however, are out in the cytoplasm, or they may be on, or they will be found on another organelle, the rough endoplasmic reticulum that I'll draw here. Good. So the ribosomes can be there. But either way, the ribosomes are not in the nucleus. So transcription and translation would not be happening at the same time. That's how I know that that is a prokaryotic cell. So as we see, multiple polymerases are transcribing the mRNA off of this one DNA template. You can also see that multiple ribosomes are then in fact binding that mRNA and going to translate that into protein, which will be the topic of our next video. Now, one thing we need to consider before we talk about translation is, so in prokaryotic cells, as we saw, the translation can begin occurring actually while the mRNA is still being transcribed. However, in eukaryotic cells, that mRNA transcript actually has to exit the nucleus before translation can begin occurring on the ribosome. So the mRNA needs to mature, or it needs to be processed before that can happen. And so here are some things that need to occur. So first of all, there are some stabilizing proteins that will bind to the mRNA as it's actually being transcribed. Now, what do I mean about stabilizing proteins? Well, mRNA is actually is not a very hardy molecule. It is easily degraded. So these stabilizing proteins are going to help prevent this mRNA from being degraded. Also, there will be one nucleotide cap that gets added to one end of the mRNA transcript, and then there will be multiple adenines added to the other end. So we call that a poly, meaning many, poly A tail. So there's a cap on one end and a tail on the other. What are those for? Well, they either can help with preventing it from being degraded, helping it to exit out of the nucleus, or helping a ribosome to recognize this mRNA transcript so that translation can occur. Finally, something occurs with eukaryotic mRNA that doesn't occur in the same way with prokaryotes, and that's called splicing. In an mRNA transcript, there are two different types of nucleotide sequences. One is called an intron, and the other is called an exon, or areas, I would say, of the transcript. So the introns are actually spliced out and removed, and then what those exon pieces are actually put together. So the way you can remember that, the reason it's called an exon is this is what's being expressed. So when we say a gene is expressed, that means the gene is turned on and the protein product is being expressed. So the exons are those that are actually being expressed. So in this figure, we see the splicing occurring. So right here, this would represent, up here would represent right after transcription has occurred, and you see that we have multiple exons and introns that are present in that transcript. So in processing, what would happen, we mentioned that a cap would be added, and we mentioned that we'd have this poly A tail. What you see is the intron sequences are actually spliced out and removed, and then the exons are put together. Now we have what we would call a mature mRNA sequence or mRNA transcript that is ready to leave the nucleus and be translated. And we will talk about translation in the next video. We're going to practice our transcription at this point. So if this is the DNA template strand, then what I want you to do, so the underlined regions, okay, let me see this. Underline equals intron. And if it's not underlined, that equals exon. So what I want you to do is I want you to figure out what would this sequence look like. First of all, I want you to tell me what is the mRNA transcript before it's been processed or before it's been spliced. Then I want you to tell me what the mature, mRNA transcript looks like. So pause the video in just a second, write out the sequence of what the mRNA transcript looks like, and then write out, after you splice it, what the mature one looks like, and then come back to the video and see if you're correct. So what I'm doing is I'm just determining, right, if I see a thymine here, when the RNA polymerase reads that, what nucleotide in the mRNA transcript is it going to add? And so that's going to be an A. Okay, this is not going to be a T because RNA doesn't have T. It's going to be U for uracil. Okay, G, A, U, G, A. So hopefully that's the sequence that you determined in your mRNA transcript. Now what I need to do is make sure that I carry out or designate in some way what is the underlined area. So this area right here is underlined and this area right here is underlined. So I'm going to get rid of these, right? These are the introns that get spliced out. So my mature sequence then, I carry the rest down. I'm going to skip this underlined sequence. And skip this underlined sequence. So hopefully that's what you obtain from your mature mRNA. Now, we also would need to add a cap, right, to this end, and this would have a whole string of adenines to this end of the molecule.